And I want to thank Sashmita, Ali, and Farhat again for inviting me as a keynote speaker. It's always to give talks without traveling, so I really appreciate that. So I'll start with a bird's eye view of where I see the research in my group in the field of genomics, and then we'll go from there. So, as you know, with now our fancy gadgets, we can take all sorts of phenotypic measurements on humans. Every time you get an Apple Watch update, there's now a new type of measurement that it's getting on you. And then in parallel to this, with the advancements in biotechnologies, we can make very precise and high-dimensional measurements at the cellular level. Of course, what's really interesting is to be able to understand how these measurements that are at the cellular level relate to each other, and further how we can use these measurements at the molecular cellular level to infer what might be happening at the organism level. So my group generally works on this area at the molecular level with hopefully obvious links to what's happening at the phenotypic level. So the two vignettes that I'm going to talk about today is about single cell data, which now has been around more than 10 years. And as you know, while initially most of the efforts, most of the technology was robust for transcriptome profiling, now we can actually robustly profile multiple modalities. So I'll give a very brief review of what type of data types I'm thinking about when I talk about single cell data. So as we all know, single cell RNA-seq profiles at the cell level the gene expression and generates this nice, neatly structured matrix where cells are, rows are cells, columns are genes. And then the second most commonly generated data type with single cell technologies is chromatin accessibility, which is a type of epigenomic mark where again we have the cells, but now, as we heard yesterday, there are no fixed experimental units. Usually the data is summarized at the level of peaks. These are short regions where at least in one cell there is some regulatory activity. And the third data type that I'm thinking about, which is far less common in the public domain, is single cell high-C data based on high throughput chromatin conformation assays, which essentially quantifies the proximity of the loci along the genome in 3D. And this data type generates a matrix for each cell. And this is, of course, incredibly important data modality to understand how gene regulation, long-range gene regulation happens. So all of these are molecular phenotyping. And in addition to these, at the cellular level, we can have electrophysiology, morphology, among many other cellular phenotypes. And especially for neuronal cell types, these are very important phenotypes. So the first vignette that I'm going to talk about, this is going to be, so I know Kwang-Moon I heard that he gave a really good talk, great talk yesterday. It will be a little bit of a repeat of that. So if you have more questions on that talk, this will be your opportunity to ask those questions, and I'll present some of the results that Kwang-Moon didn't have time to cover. And the second part, we are going to talk about bringing data from different modalities together. Okay. Okay, so in addition to profiling molecular events in the cell in their native state, of course, with now CRISPR technologies, we can induce perturbations in the cell and then profile various molecular events after these perturbations. And a great, one of my favorite papers from this area is this paper from 2 years ago that focused on MYC gene, which is an oncogene, very important for cell proliferation. And the activities of this gene is controlled by 4 enhancers that are immediate downstream of the gene, and another 3 enhancers that are very far away from the gene. Okay. So this experiment, the design utilized multiple guide RNAs to interrupt either individual enhancers one at a time or combinations of enhancers. So the idea is to really understand what the effect of these enhancers are on the expression of this gene. So what they were able to show that if you consider two enhancers that are proximal to each other, that are nearby each other, if you disrupt one of them, or if you disrupt both of them, the effect can be explained in an additive way. So as you see here, if you delete E1, one of the enhancers, MYC expression goes down a little bit. If you delete E4, it goes down a little bit less. If you delete both of them, it goes down more, but in a way that's consistent with the additive model. Okay, same for enhancers that are farther away. These guys, again, they're farther from the gene, but in close proximity of each other. If you delete one of them, it goes down a little bit. The other one, it goes down a little bit. If you delete both of them, it goes down in a way that's consistent with an additive model. Okay. However, if you take an enhancer from this region and an enhancer from this region, the expression goes down in a way that's not consistent with the additive model. So this suggests an interaction between these two enhancers. So if you think about this, is of course incredible, and it opens up a whole new type of analysis for molecular QTL and GWAS type studies. Because if we have SNPs disrupting one enhancer here and another SNP disrupting one of the enhancers here, then you could presumably argue that maybe there are interactions, maybe you would see interactive effects of these enhancers. So after this paper, we were actually motivated to see, okay, maybe there is a way to reduce the whole space of SNPs to look for interactions by first looking for whether given regions are physically interacting. So that's not going to cover all sorts of interactions, but that's going to be a first step in the right direction, because at least any given region any given regions are physically interacting. That could imply that they might have downstream interactive effects. So essentially, we are thinking of, we call these multi-way interactions that have at least one gene and at least two enhancers. It could have more genes, more enhancers. And not so surprisingly, in the last several years, there has been a lot of technological advancements in profiling these multi-way interactions directly. So there are a lot of them published in high-impact journals, but mostly I would say they are at the proof of principle stage. They are not widely adapted yet. So there's no winner-takes-it-all method out here yet. So it's fair to say that they are still in the developmental stages. So then we ask, perhaps we can infer multi-way interactions with a more abundant and robust data type. And the most natural one to look at is high-C data, and particularly single-cell high-C data, which captures pairwise interactions. So I'll skip these. We heard a lot about high-C already, but at the end of the day, you get a contact matrix. And with the availability of single-cell profiling, we can actually do these experiments at the single-cell level. And then for each cell, get a contact matrix that quantifies the interactions, long-range interactions in that cell. And currently, for the Nucleome and NIH BREAM initiative are two entities that are generated this type of data. So the high-C data can be nicely organized in a tensor structure. So it's a three-dimensional tensor. One dimension is cells. You are stacking up the cells, and for each cell you have a matrix. This opens up nice tensor-related, high-dimensional tensor-related problems. And Kwang Moon in my group worked on these. There are both supervised and unsupervised problems, because once you have this data, there are many different types of downstream analysis that you can do. So I'm not going to focus on them, but this work is published and also out there in the iCarve. If you're interested in these type of problems, I would say these are good manuscripts to start with. Okay, so now going back to whether we can use single-cell Hi-C data with quantifies pairwise interactions to learn about multi-way interactions, we had to think a little bit about this problem. So in a given cell, if you have a three-way interaction, including, so here in this graph representation, each node is going to be a locus in the genome. So if you have a three-way interaction, so at the pairwise level, you'll see all the pairwise interactions. So then if you think about single-cell Hi-C data, that's great news. But then if you think about how this data is generated, there's a ligation step that randomly ligates the fragments that are in the interaction. Actually, it's very unlikely that you are going to see all the pairwise interactions, especially as the multi-way interaction gets larger in the number of genomic locus it includes. So for example, in this case, you might end up seeing this purple and pink, and then pink and green, but not this one in a given cell. But then if you think a little bit about it, so if you think about it as a, if you're expecting that you would see all the pairwise interactions, then this is almost like finding cliques, fully connected subgraphs in a graph, but the nature of the Hi-C data is very unlikely to provide that. But then the good news in a way is that if you're looking for multi-way interactions, you don't actually have to see all the pairwise interactions in there. Right? So for a three-way interaction, if you have this and this, then this already implies that this interaction also has to be there. So that's the realization. But then, of course, we wanted to see whether if you actually look at the real data at various resolutions, whether you are seeing full cliques, right? So we did that at the, so some of the cell line data that's very low resolution, but the data that's coming from the Brain Initiative is a much higher resolution. And we can see that there are a lot of actually not just pairwise interactions that are occurring simultaneously, but actual full cliques. So this gave us the confidence that this is probably a viable task. And Kwang Moon talked about this yesterday, so I'm not going to go into the model details, but we have a generative model that for the Hi-C data that generates pairwise interactions according to a null model. So this takes into account potential technical biases in the data. And then at the end of the day, it quantifies the strength of the pairwise interactions. So, and then the process becomes, how do we aggregate these pairwise interactions to claim that we are observing a multi-way interaction? Okay. So given candidate interactions, these are cliques from the actual data, we realize that you don't have to observe all pairwise interactions, so you just have to observe R of them, some R of them. And so we, so in a way, so if you are looking for, if you are requiring R of the interactions to be there, then one natural thing to do would be, if you are thinking about pairwise interactions quantified with some p-value, I'm going to look at the Rt smallest p-value. Or if they are quantified as z-scores, I'm going to look at the Rt largest z-score. So we are looking at the Rt order statistics of the actual statistics we generate at the pairwise level. So this is a very intuitive idea. And then the nice thing is that if you consider Rt order statistics, if your p-values are well calibrated, then actually the Rt p-value is going to have a beta distribution. Okay? So this really is the idea of the aggregation for these pairwise interactions. And then, of course, what is R? Kwang Moon did a lot of empirical experiments, and it turns out that if you set R as the size of the clique -1, that gives really nicely calibrated p-values. Okay. So, as most of you know, most of the actual work in this area is done for validating somehow what you identify, and we have done a lot of experiments for that. We looked at mouse and human data, cell type data, data from cell lines at different resolution. And we used essentially all the relevant data types that we can get our hands on to convince ourselves that what we are getting is something reasonable. So you already saw some of those results yesterday. I'm going to show you some. So we think we are the first ones to ask this question to the best of our knowledge, asking, can we really infer multiple interactions from single cell Hi-C data? So we designed a simple baseline approach. There can be other baseline approaches that some of you might come up with. So essentially, we took what I would call state-of-the-art method for identifying significant pairwise interactions from single cell Hi-C data. So that's this SNAP Hi-C, commonly used by the Brain Initiative. So you can apply that and learn all the pairwise interactions at some FDR cutoff, and then you can aggregate them into multi-way interactions. So that's what we did in a simulation experiment where we simulate actually utilizing actual observed data and then implanting multi-way interactions at various strengths. So we can see that the aggregation approach has overall very high false positive rate. So our approach, which we call MIN-C, has a really good control on false positives. And then this comes at the expense of course, being underpowered where the multi-way interaction signal is low, but it immediately catches up as the multi-way interaction becomes stronger. So ideally, so this aggregation that we are using, this order statistics, it just depends on your p-values. So we have one way of generating those p-values. So we were curious why this idea wasn't really working for SNAP Hi-C. And it turns out that SNAP Hi-C p-values are really not well calibrated at all. So this is actual data. So this is expected p-values. If your p-values are well calibrated, as you know, most of the data should follow the p-values should follow a uniform distribution and some variation from it. Whereas if you look at the pairwise p-values that come from MIN-C, they are well calibrated. There's some very high signal, so you get, there's a chunk of the p-values that deviate from the expected distribution. And then also this accordingly at the clique level, when we are evaluating multi-way interaction, gives us well-calibrated p-values. So I'll show you one evaluation that I don't think Kwang Moon has shown. So we focused on multi-way interactions from prefrontal cortex, and then use site encodes resource that quantifies gene enhancer interactions. So we took all the multi-way interactions identified by MIN-C, our method, and this summarized approach by utilizing state-of-the-art interaction calling for Hi-C data. So we took all these interactions which have one gene and multiple enhancers, and then we asked whether if you have a gene in a multi-way interaction, whether the other enhancers are supported by the site encodes gene enhancer promoters. So overall, we see that MIN-C has larger proportion of validated G by enhancer interactions in 6 of the 8 cell types. So this is just one of the validations. It has a lot of drawbacks, but with the additional data you have seen yesterday, it's giving us good support that these interactions are real. So of course, we want to ultimately be able to use these interactions in molecular QTL or GWAS studies. So towards that end, we looked at an Alzheimer cohort, an eQTL study of Alzheimer cohort, and the eQTL study was done in cortex. So we looked at our multiple interactions that we identified in the cortex, and we took all the interactions that included at least one gene. And then we looked at whether the other loci in the multiple interactions harbored SNPs, and there were about 321 of them. And then we evaluated whether the variation in the expression can be explained with an additive model or with a model that has the interaction. Because that's what the state of the art for GWAS is. Even with very large GWAS, with almost 1 million sample size, we just looked at additive models. So this is reducing that space immensely, of course, right? So from the millions of interactions that I can look at, I'm only here I'm only allowed to look at 321 of them. So then this red line is the QQ plot of the p-values from an f-test that tests for this interaction. And the gray line is the QQ plot of the p-values you get if you permute the data, break the link between the expression and the genotype. So as you can see, the interactions are deviating clearly from the model that captures from the null model, and actually a number of them survive multiple testing. And an interesting example is this DKK3 gene. So we identify an interaction that included the promoter of this gene and two of its introns. And interestingly, these introns harbored regulatory regions based on some external data from cortex, and then they harbored these SNPs. So if you look at, so when you evaluate what happens to the, so this is adjusted expression because there are so many other covariates in this study. If you have only minor allele from one of the SNPs, that's what the expression looks like compared to having no minor alleles. And then if you have the other minor allele, so it's not even significant. But if you have both minor alleles, then there's actually a real significance from not having any minor alleles. So clear support for the interaction between these two SNPs. And interestingly, so this is a work, we are doing more follow-up work on that. Both of these SNPs actually disrupt transcription factor binding sites, potentially suggesting that two transcription factors interacting, coordinating the expression, and then when both of them have disruption in their binding, that has potentially higher impact on the expression. Okay, so that concludes the first vignette, and then now I want to focus on once we have these very different modalities from single cell technologies, how do we actually integrate them and then impute the missing modalities? So this is the work of Sichi in my group. I believe he has a poster that I hope you get the chance to see. So now these single cell technologies are really good at many different single modalities. RNA and attack, I think that data, many labs can generate that kind of data, and even methylation, 3D genome. So we are pretty good at generating all of these. And of course, there are even technologies that can generate multiple modalities. But ideally, where we want to go is for a given cell, we would want to see all the modalities. Okay? So then the question is, so we might eventually get here. Obviously, there might be some magic technology which gives us a lot of modalities, but there is already a lot of data in the public repositories. So there are already some rare samples that have been, profiled for RNA and attack. So eventually, you would, if you want to study those rare samples, you would want to be able to predict these other modalities, impute these other modalities for them. So more explicitly, the problem is you are given a group of cells with certain modality. This is maybe RNA, attack, and then another group of cells with another type of modality. And then obviously you want to first integrate these somehow, and then also predict these missing modalities. So integration is a very loaded term, and it means different things to different people. So in this domain, the very basic integration, I think this audience this is probably very obvious to this audience, is very basic integration is if you have the same experiment done by different labs or different times or different machines, and then if you do nothing and then combine the data to increase the power, then you are going to see that your cells are going to be clustered based on the data source, which lab they are coming from. So then you want to do something so that when you cluster your cells, they are clustered by the cell type. So that's the very basic level of integration. And then the second level of integration is, okay, now maybe you have a modality which doesn't exactly quantify the same features. So you have RNA modality, which has genes as the features, and then you have attack modality, which has peaks as the features. So here, people have convincingly shown that if you can summarize these features somehow, if you can transform these features to the features in the first modality, then you can still integrate them. So this works really, really well for RNA and attack. And in fact, we were able to show that you can play the same game for high-C data. We developed this quantification method, that's single-cell gene associating domains, which quantifies all the interactions that originate from the gene. And then you can summarize your high-C data as cells by genes, and then use any type of integration method to integrate these two modalities. So I have this idea in action where we are looking at a prefrontal cortex data. So RNA-seq from a 2021 paper and high-C from a 2019 paper. So this is the integrated data. The labeling indicates the data source. This is perhaps a better view. The colors indicate whether it's high-C or RNA-seq, and we see that there's really nice integration. And then in this case, integration really helps to separate the cell types in the high-C domain, which is much more sparse compared to RNA-seq domain. So in this specific example, it helped us to separate the excitatory and inhibitory neuronal types for high-C cells. So there's, as you can imagine, this is a very, very crowded area. And it's also not, I wouldn't say, it's also not very easy to classify all the work out there in a fair way. So we have been thinking about how to do this. And I think what's useful is to think about an axis that's focused on data integration, and then another axis that's focused on multimodal prediction. And we have done our best to place these methods here, but this is going to change as we learn more about these methods. I actually tried ChatGPT I gave it the papers. I'm like, okay, I want them to be classified along a data integration axis and multimodal prediction axis. And I can tell you that it's not as good as me or Sichiyet, so that's good news. But anyhow, so there's a lot of work in the integration domain, a lot of them, and there's now new work, like this SC butterfly is from this June or August. It's actually really new. So then in principle, there's a lot of work in integrating RNA-seq and attack-seq. So our goal is, can we start integrating data from other modalities? Patch-seq, high-seq, that really is the goal. So we said, okay, maybe we take some of these methods that are down here and then see, what the state of the art would be if we applied these. So we did that for patch-seq data. So patch-seq, it's by definition, it's multimodal. It profiles electrophysiology and also transcriptome, but much smaller number of cells because patch clamping is a very laborious process. So it would have a few thousand cells. And then we took high-C data, summarized at the gene feature level, and then applied some of the much newer methods like MaxFuse and MIDAS, and also some of the really robust methods that are widely used. And that's essentially what you get at the integration. So it's not for Seurat and Liger, maybe it's not that terrible, but there's a lot of room for improvement. And then again, furthermore, as I said, so with these methods, the methods that focus on integration, if they can integrate, usually imputation is based on some sort of k-nearest neighborhood. So that's the natural thing to do. So we also wanted to see whether we can push the limits on this axis, on the multimodal prediction axis. And so I've been emphasizing the structures of these data. So these are mostly matrix formatted data. So that immediately brings to mind, mostly unsupervised methods that leverages varying complexity autoencoders. And most of the methods on that graph, if they're not using basic statistical approaches like canonical correlation analysis, they rely on autoencoders. And dual autoencoders, but they rely on the matrix structure of the data. So we said maybe we want to first convert the data into a graph, okay? Actually into a heterogeneous graph where the nodes are cells and also the features. So in the graph, you'll have some nodes that are cells, some nodes that are genes, and some nodes that are peaks. So in the machine learning literature, that's called the heterogeneous graph. And then if we can do that, because then all this multimodality, missing modality prediction problem actually becomes a link prediction problem, which is, there's machinery, a lot of machinery, abundant machinery at your disposal when you are at that stage. So this is how we construct the heterogeneous graph. First we have some observed links. So these circles are going to be cells. So we link each cell to different gene nodes. So, and then the edge weights represent the expression of the gene in that cell. Okay? So that's the easy part. Okay? And then, of course, we can do within modality cells. So within modality, we can calculate distances between the cells and then link cells to each other. So this is also kind of nice because, from a statistical point of view, if you have homogeneous units, you somehow want to be able to borrow information across them. And if you are purely treating the data as matrix and feeding it into an encoder, perhaps there are less opportunities to do that. But once you link the cells based on their similarity in this domain, now there's going to be more opportunity to borrow information. And then finally, we want to link cells across modalities. One can use very different things here, but Seurat is quite robust, and so we use the canonical correlation type approach that's actually also implemented in Seurat. So once you have this graph, so then all you have to do is run a graph neural network on this and learn lower dimensional representations, node representations, for both cell nodes and also feature nodes. So now everything is represented in the same space. And that way, this is going to enable, if I want to predict or impute modality, like green feature modality for this cell, that's going to be a link prediction problem. Okay, so this is Sitchi's figure, much nicer. Input data turns into a graph, and Sitchi actually runs something called MetaPath2Vec, which learns cell embeddings that are used to initialize. And then once we learn these embeddings and the functions to predict links, then you can use this to do prediction, denoising, and other type of inference. Okay, so we did a lot of computational experiments using patch-seq, site-seq, and then attack and RNA profiled together so that we have some sort of ground truth. And in terms of the cell settings, we had this setting that allowed partial overlap. So for a subset of the cells, we had both modalities measured, and this is mostly because a large fraction of the methods that exist on that axis that I showed you require that you have some cells with both modalities. And then also looked at setting where there is no overlap between the modalities. Okay, and in terms of the results, once we have, on the test data, we have the observed quantities, we predict them. We looked at correlations across cells and also across genes. So I have some results here. So this is a setting, this is an experiment on the actual PATH-seq data. So I have a few methods represented here. These are some of the newer methods that can do integration and multimodality prediction. And then we also threw in a KNN just to see, once we have our heterogeneous graph constructed, what if instead of running a graph neural network, we did something much more simpler? We just did KNN for imputation. And as you can see, it actually does pretty well. So this is cell correlations and also feature or gene correlations. And then overall, our approach that runs graph neural network on that initial graph is better than KNN in about 80% of a little less than 80% of the cells. So this is a glimpse of what the integration looks like with methods that actually generate embeddings. And then this is the evaluation of the integration performance. And then we see that Gleam does a much better job of mixing data, cells that are coming from different modalities, and then separating the cell types. This is the prediction performance. Overall, Gleam does better, and then seems to still have an edge on KNN. So then, of course, these are just, we can calculate many other metrics, but at the end of the day, we actually want to look at, pick one cell, pick one gene, and then look at what actual predictions look like, right? So because here there's so much stuff going on, so the data is summarized across many things. So here we are looking at a single cell, and then we are looking at how its expression profile is predicted. So x-axis is observed expression for the cell, y-axis is the predicted expression, and then these are what the predictions from different methods look like. So most methods struggle with zeros. That's very natural, and Gleam uses a hurdle model in the sense that it first tries to predict whether there is a link between a cell and a gene, or a cell and an attack feature. And then if the link is there, then it tries to predict what the actual value is. So this is attack predict. So this is one peak, one genomic region, and its accessibility. Oh no, this is one cell. Okay, this is still one cell, and then its accessibility across all the peaks. And then we again see methods struggle with the zeros. CMODs developed by my colleague Daifan's group, it actually does pretty well because it really focuses on integration, and then the prediction relies on KNN, as far as I can understand. So this is an example. Now the modalities don't have any shared cells. SC butterfly is a recent method that's published, and then we can see that it doesn't do much better than KNN. Okay, so this is more or less just to have an idea how this method is performing. We will do more experiments. There can be more scenarios that we want to investigate these methods, but ultimately we want to use this to see if we can actually combine patch data with high-C data. So we did all these experiments where there was ground truth, because there were cells where both modalities were profiled. So first, so this is more of a real application of Gleam. We trained it on the PATH-seq data, and then we took atlas level RNA-seq data just to see, the electrophysiology is trained on this data, how well we can predict it on the atlas level data. So this is showing for each cell type what the electrophysiology looks like. And as you can see, it captures the overall trend pretty well. And then, of course, the next thing that we want to ask is whether it's actually capturing the variability. And as you can see pretty clearly, so the variability in the actual data is larger. For most cell types, is larger than the variability in the predicted data. So I have a better quantification of this. So for each cell type, so this is the variability in the patch data, and then this is the variability based on the predicted data that's using the atlas level RNA-seq data. And then from this, we dived into L5 a little bit better. So we looked at the cells in the GLEAM embedding space. So this is what they look like in the patch, and this is what they look like in the atlas level data. And you can see that L5 is actually not just one cell type, it has subcell types. And it turns out that one of the subcell types, L5MP, is actually barely in the patch-seq data. So things like this are, of course, contributing how well we can capture the variability, but nonetheless, it captures the fact that electrophysiology for this one particular, I can't remember. So one particular, this upstroke to downstroke ratio, that's one of the electrophysiology features that's very different between L5IT and L5ET cells, and that's something known in the literature. And another, again, more realistic application of GLEAM is taking GLEAM to integrate patch-seq and Hi-C data. Here again, patch-seq is 2 modalities. And then Hi-C data, this is one of the Brain Initiative data sets. We have 3D genome, but also methylation. So we are trying to integrate 2 different data sets, each have 2 modalities. So if we can do this integration, of course, then we can ask whether electrophysiological features can be, variation in the electrophysiological features can be linked to 3D structure of the genome. So this is an analysis towards that direction. We essentially sorted all the cells based on these cells, based on their predicted electrophysiology feature. So that's the top row here. And then we just let the regions of these cells cluster based on their 3D structure. Okay? So when the clustering is happening, it has no idea about these electrophysiological features. But as you can see, it's naturally generating a clustering that correlates with the heterogeneity in the electrophysiological features. And furthermore, if you label the cells, if you look at the labeling of the cells, it's actually the first group is excitatory neurons, and then it's giving us, with 3D genomic regions, have to have certain type of signal for excitatory neurons. So these are the type of analysis. This is very basic analysis, but these are the type of analysis that we are going to be able to do once we are able to integrate these data types. Okay, so I'm going to summarize here. So in the first vignette, I talked about identifying multi-way interactions. I think there is a lot to do in this area. So we have a basic idea of how we can aggregate pairwise interactions. But for those of you here who are more comfortable with hypergraphs or other type of machine learning machinery, I think there are a lot of opportunities here. And then in the second vignette, I hope I'm able to show you that once we can integrate these different modalities, we can actually start asking more elaborate questions about how phenotypes at the cell level relate to molecular phenotypes of the cell. Okay, so with that, I'm gonna again acknowledge Kwang Moon, who led the first part, Sichi, who led the second part. Both of them are looking for postdocs. So if you or your colleagues are looking for postdocs, they're both fantastic. And then I want to thank the rest of my group. We collaborated with Ye on parts of this work. And then when we started thinking about multiple interactions, we weren't even sure whether this is something viable. So we talked to a lot of people who generate this type of data, and I want to acknowledge that too. So with that, I'm happy to take any questions you might have.
Hi, fascinating talk. If you were to use that inference logic on bulk IC, where you would be integrating across different cell types, what percentage of bulk multi-interaction do you think happens at the single cell level? And also, when you look at the context between these regulatory elements, the majority of them seem to happen between enhancers without promoter participating.
Yeah.
Do you think that interaction has any functional meaning, or is it just noise?
Yeah, okay. Very good questions. So the first question is, so I think to identify multiple interactions, actually, you don't necessarily need IC data. If you have purified cells and you have a method that can generate, well-calibrated p-values for that bulk IC data, you can probably identify several of these. You can probably identify a good portion of these multiple interactions. The tricky part is, though, when we are doing this, we start with a set of cliques that exist in at least a small number of cells. Okay? So these cliques need to appear in at least 2-3 cells, because otherwise you are left with, everything can be, if you are constructing them from pairwise interactions, the space is just big. So here, having single cell data is really constraining us, because we know that, okay, this candidate clique, we see that in at least a small number of cells. Okay? So that's our starting point. So then we evaluate that. So you could, for example, that's an experiment we haven't done. You could take candidate cliques based on single cell data, and then go to bulk data, and then evaluate, calculate some pairwise interaction. If the p-values are well calibrated, then you can use MinC the way that we are aggregating. So that's number one. And the second thing is, since the single cell data is there, so I don't think we need to bulkify. Okay, so that's another reason, because we have the replication, we should use it, get a better p-value out of this replication. So those are the two remarks I want to make. And so I don't know, so we haven't really done that experiment, like take your cliques from single cell and then, look how much of that you are able to recover from bulk. My concern would be that the p-values from bulk data are not going to be, based on our experience, are not going to be well calibrated. So that's number one. The second question was, most of the interactions are not between genes and enhancers. So if they are, interactions with lots of non-coding regions, do they mean anything? I would say not necessarily. If you look at data from Sprite, GAM, which directly profile these multiple interactions. I mean, they have these interactions with, thousands of loci in them. So I don't know what it means. I mean, the cell is small, so sometimes everything needs to touch each other. But I think it's reasonable to argue that if it involves genes and enhancers, then that is a mechanism for actually interacting in a way that can affect molecular outcomes.
It's really great methods. And what I was wondering for the patch-seq data, have you ever thought about the contamination of the RNA from the RNA component? Because when they're doing the patch clamp, the mRNA from the adjacent cells can be brought into the cell which you're using it to further measure the RNA.
Yeah.
And in our study, we find that there are like some contamination and also got confirmed by Allen Institute. So I was wondering.
I believe that this is Allen Institute data. RNA sequencing from patch, that's lower quality for sure. And that also makes the integration harder. Yeah.
Yeah. I was wondering if you kind of like cracked the contamination, would you get like a better prediction score?
We haven't done that, but yeah, I think if we improve, because when you have patch-seq, the initial graph construction is really relying on RNA component and whatever component you have on this modality. So I think it has potential to improve. Yeah.
Thank you.
Sorry, guys. My original question was actually answered in the first.
Good.
But I have another one, if you don't mind. So I was wondering, for the prediction from one modality to another, if we assume that all information for one modality is already contained in another modality, then why even measuring the other modality, right? So I don't think this is the case, obviously, but I'm wondering, by developing this predictive model, is there any way to interpret them so that we can try to understand some causality in the regulations between modalities? Maybe is there any thought about that?
Yeah, I think you're asking a very good question. So everyone was integrating and predicting the different modalities. So we said, why not? Let's see whether we can do it. But I think, so first of all, before I think we can get there, to really learn real biology, I think we shouldn't be satisfied with just a single number for prediction. So our next step is to do, to get confidence intervals on this, to do conformal prediction, so that we can really be confident. And then I think we are going to identify that for some cells, actually the predictions are really bad. The confidence intervals are wide. So that could indicate that maybe there are different cell types or cell states. So I think there is really potential to identify those type of things. And then other than that, so Hi-C is kind of out there, 3D genome. It has more information than what ATAC has. So I think integrating it with patch is interesting, because you can imagine that the size of the cell might have something to do, how efficient it is in its electrical output. So the premise is there, but I'm not sure whether, we don't have any new biology yet, let me put it that way. But I think there is potential for it.
Right. Thanks a lot for the presentation.
Great talks and parts as usual. So I had a question about the MINT-SE part. So you're not pseudo-bulking. You're calling interactions at the individual cell level, the single cell Hi-C.
We are not identifying interactions for a given cell.
Okay.
So we are using cells as replication so that we can quantify those pairwise interactions really well. But at the end of the day, we are analyzing a single cell type, and then we are saying that these are the multi-way interactions for this cell type. So you would expect that a majority of the cells actually have that multi-way interactions.
But where are you starting from? What is the pairwise interaction coming from? But anyway, I guess that's a small technicality we could discuss later on.
Okay,
what is the resolution that you're looking at? Like, how big are the interactions? How big are your cliques? And can you look at TADs to restrict it in some way? How much of the genome is a clique actually covered?
So first of all, data is sparse. So most of the multi-way interactions are 3-way. So we can get some 4-way, some 5, 6. I think it tapers off completely at 7. So that's number one. And number two, the best resolution we have now is 10K. So that's actually pretty good. Yeah. Of course, we had to do experiments where the resolution is, half a megabase. And also, like sprite interactions are also like that. You know, the existing technology also can measure. It doesn't have better resolution anyway. But of course, you know, when the region is so big, so then it's like interaction between the tags that would still be meaningful, but, you know, like using it for molecular QTL or GWAS type of analysis, I mean, that just wouldn't be realistic. But with 10 kb, I think we have a good entry point to being able to do this. Especially you have your, you know, in the example that I showed, we have 10 kb, but those regions actually have regulatory regions, you know, with attack and other histone signals, and the SNP happens to be there. So there's really good additional data that can be utilized to filter multiple interactions that are, you know, not probably going to be useful, but then hone on the ones that, you know, that might get a signal.
Very, very interesting.
I'll ask one question. Someone stole his question. He stole my question, I think, in a way. But my question was similar, but coming from a different angle. In terms of like these, you know, imputation methods and ways of like finding the missing modality. Sometimes I feel like they're very sophisticated ways of learning cell type or memorizing cell type information or cell subset information at the end of the day, right? But I think maybe you had some results where you're working with like a single cell line and single cell data out of it, or like, are there cases where within a given cell line or a cell subset, these imputation methods are able to capture that variability so that we believe that there is really something like what you're saying, like your motivation, that 3D of the same type of cell, the 3D organization of it has something to do with electrophysiology.
Yeah, so even in the electrophysiology example, right? So the imputation on the test data, this atlas level data, so the trend is there, but you could say, I don't need the trend to be there because my atlas data, I identify cell type and I'll have a, you know, I'll have a mean value for it. But we get pretty good variability on the atlas level data. It's not perfect. We are still underestimating the variability, but it's actually pretty good than, you know, just using the mean.
I see. So, but if you took one of those specific, like if you took L5, but then L5 also got divided into 3 and all of that, if you got that one subset of the L5 and then looked within that, then per cell predictions, are they like, is there some relationship between those two different modalities that, in terms of growth?
Yes, so that's right. So that's the analysis where we predict, for example, we predict electrophysiology even for a cell type. So yeah, I didn't show some of the results we have. There is great deal of variability, so predicted variability. And then you can take, you know, 3D features from that cell type and look at the association. Yeah, we are finding a lot of association. And we can do this, you know, associate electrophysiology with 3D features, electrophysiology with RNA, and then we can, you know, there has to be some correspondence, you know, what we are learning from RNA world and the 3D world, there's great correspondence, yeah.
Thank you.